Okay, I'm just gonna read it once.
Did it.
Amazing. Yeah.
Hi, my name is Christina Buselli. I am a research associate in the Finkbeiner lab. I came to Gladstone after finishing my undergraduate degree at the University of Portland for the opportunity to advance my basic research skill set while also being able to still be involved in an area of research that I already had some background in. In college, I was involved in a project that that tried to verify the suspected causative mutation for lysosomal storage disorder called GM1. This was my first exposure to studying lysosomes and their role in neurodegeneration, and I wanted to continue studying that after college as well. And serendipitously, I was able to join the Finkbeiner lab, where I'm a part of a team that studies a different neurodegenerative disorder called frontotemporal dementia, which still also involves studying lysosomes. I really admire the humility and perseverance of my coworkers at Gladstone. I think Gladstone is a place where it's okay to learn from mistakes and where compassion for others drives hard work even when no one is watching. I also really appreciate the opportunity to learn from people with different skill sets and people from different backgrounds at Gladstone. For example, I love learning new lab techniques from the biologists and RAs in my lab, but I also love learning more about data science from the software engineers in the Finkbeiner lab as well. I'm a part of a team that studies an early onset form of dementia called frontotemporal dementia. And when people see the word dementia, they often tend to associate it with memory loss, but frontotemporal dementia also involves progressive changes to behavior, personality, language, and movement. And we're particularly interested in a form of dementia called progranulin deficient frontotemporal dementia, which has a decrease in the secreted protein called progranulin. And we study how this decrease affects all sorts of things in different central nervous system cell types like astrocytes, microglia, and neurons. We are particularly interested in how this decrease impacts lysosomes, an important organelle that helps to degrade certain molecules that could otherwise become toxic if they accumulate in cells. We are also taking it one step further and looking into the downstream effects this decrease in progranulin has on other proteins. To do this, we essentially start by growing cells like microglia, neurons, and astrocytes, and then we stain those cells with markers for these proteins of interest, and then we image those cells to try to quantitate and look for different types of phenotypes. I actually took a pause from studying science in college for a while, but I was eventually able to find my way back to it thanks to the mentorship I received from my undergraduate research mentors at the University of Portland. I owe it to them and all the student researchers I was able to collaborate with because they showed me what a career in science could look like, one where it's okay to learn from mistakes and where collaboration can triumph over competition. When I'm not working, I really enjoy playing the piano, running, trying to make breakfast for dinner, and reading books about bioethics. I'm actually currently working on a manuscript with some of my undergraduate research mentors about the ethical implications of gene editing technology. So we evaluated college student opinions about using gene editing for a variety of purposes, like treating somatic versus germline genetic disorders or altering non-human species for reducing disease spread, and so that's also something I work on in my free time. I would love to get better at fly fishing one day and actually be able to catch a fish.